Good evening, everybody. We're going to get started. And can I just check that everybody at home can hear me on Zoom? Great. Well, first off, I'd like to introduce myself. I'm Jane Wilkinson, the Executive Director of the Koch Institute. And I want to welcome all of our guests, both virtually and in person. I'm personally thrilled to actually have an in-person event for the first time. So this is very, very heartwarming for me. And it's good to see all of the faces in real life as well as virtually. Now, this is our fourth and final With Insight program in this year that we're celebrating our 10th anniversary. And so far, we've covered lots of different topics. We've talked about some of the unique types of research that we do here at the Koch, including the Swanson Biotech core groups. We've been talking about our clinical programs. We've also been talking about our research programs that have covered both the Bridge and the Frontier programs that we have here at the Koch. But tonight, I'm absolutely thrilled to showcase our entrepreneurial work that we do here, especially around our cancer research. We're a National Cancer Institute designated basic research center, and we have been for almost 50 years. And although we don't treat patients directly, and I think that's one of the strange yet unique things about the Koch, in that we don't actually are affiliated with a hospital, connecting to patients and developing patient therapies is always on our minds here. To do that, we rely heavily on collaborations both within MIT, but also within industry. And it's really a focus of ours to make sure that our research does get delivered to the patients. Now, sometimes this delivery is a partnership with industry, but sometimes it means we have to set up our own company. We're gonna move this innovation into a new company, and that's what we're here to talk about tonight. We're here to talk about the entrepreneurial spirit that is here at the Koch. And since the Koch was formed 10 years ago, we've formed over 100 companies. So this really is, really has got an entrepreneurial vibe here. The first half of our evening is going to be some mini presentations from some of our scientist entrepreneurs here. And then we're going to transition into a panel discussion. That'll happen at about 7 o'clock. I would like to take questions both from the audience and also through the Q&A box that's on our Zoom window as well. So with that, it's my absolute pleasure to introduce Bob Langer. And Bob is going to talk briefly on his absolute passion for entrepreneurial work. And I think Bob could really write the book on this. So Bob, I'll hand over to you.
Well, thank you so much. I'm just going to look at my notes, which is what Erica told me I'm supposed to say. But basically, I thought I'd just talk for a few minutes to tell you how I got involved in all this. So my story goes back to 1974, when I graduated from MIT and in chemical engineering, and all my friends in chemical engineering went into the oil industry because they had all these high-paying jobs. And so I thought, and it's amazing, they just were all over MIT. So I thought I would, do the same thing. So I went to these interviews. I actually got 20 job offers, 4 from Exxon alone, and I wasn't that good, but they just, had all these openings, and even if I flubbed the interviews, you'd still get a job offer. So, but when I went there, I remember one of the Exxons, they said, well, gee, if you could just increase the yield of this petrochemical by 0.1%, that would be worth billions of dollars. And it just sounded so boring to me that I remember flying back to Boston, and I thought to myself, I didn't want to do that. So I started to think of other jobs that I could do. And one of the things I did when I was a graduate student, when I was supposed to be doing my thesis, was starting a school for poor high school children in Cambridge, called the Group School. And I'm sure Bonnie Kellerman remembers that. And but at any rate, I really liked that. And one day I saw an ad to be assistant professor at City College of New York to develop chemistry curricula. And that's what I did at that school, one of the things I did. And so I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea. So I wrote to about 40 different colleges applying to be assistant professor of chemistry education, and none of them wrote me back. So that wasn't so good. So then I started to think, what other way could I use my chemical engineering education to help people? And I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. And then one day I remember going into the lab, and one of the older scientists said to me, he said, Bob, you should write Dr. Judah Folkman. He's a surgeon at Children's Hospital. And he said, sometimes he hires unusual people. He thought very highly of Dr. Folkman, but I won't say what he thought of me. So anyhow, I wrote to Dr. Folkman, and Dr. Folkman actually was doing cancer research as a surgeon. He had this idea that if you could stop blood vessels from growing, maybe that would be a way to stop tumors from growing. I thought this was an incredible idea. I didn't know anything about biology, but I think still the last time I took a course in biology was 10th grade. But at any rate, one of the key aspects of doing that was to develop a bioassay. And to do that, we had to develop tiny little delivery systems that could deliver macromolecules. All of the angiogenesis inhibitors, it turns out, are large molecules. So everybody told, Dr. Folkman was on the board of the only company in the world, Alza, that had done any work on anything like this. This was back in 1974. And he asked, they had Nobel Prize winners on their board and other in chemistry. And he asked them, could they help? And they all said, it's impossible to deliver macromolecules. It's kind of like any of us walking through a brick wall. But anyhow, it was really important to try to solve that problem. So I spent several years working in the laboratory. I found over 200 ways to get this to not work. But eventually I did. I found a way, and we were able to deliver nucleic acids. I'll mention that in a second later. But and proteins and other things, really almost any length of time for micro and nanoparticles. So at any rate, and we published it in Nature. People were super skeptical. It got hugely criticized. In fact, my first 9 grants were rejected because everybody said it was not possible. And I'm like I said, a chemical engineer. So when I applied for faculty positions, no chemical engineering department in the world would hire me. In fact, the only reason I'm at MIT is because I got an appointment in the nutrition department at MIT. But at any rate, that's a whole probably other story. But at any rate, I really believed in this stuff, and I kept working on it. And the students at MIT, even in the nutrition department, and some would come over from chemical engineering, they were excited about it too. So we did more and more work. And I worked on this for about 10 years. And I thought, you'd publish papers and people would just use your stuff. But that didn't turn out to be true. And so but after 10 years of working on this stuff, and we had patents and things like that. In fact, by the way, I actually used some of the reviews that people wrote about how bad we were to get some of our patents, because that showed it wasn't obvious to do this. It's true, actually. I did. So at any rate, it's a good tip. So at any rate, finally, after 10 years, I got a call from a multi-billion-dollar animal health company, and they said, boy, we read your papers. We'd like to have you consult for us on to deliver an animal growth hormone. And they said, and I mentioned the patent, and they were very excited about it. And they said, well, gee, we'll give you a consulting fee, and we'll give you a $200,000 grant, and we're going to work on this. I thought, this is incredible. I went from a Ford Pinto to a decent car. And so anyhow, my Ford Pinto was like falling apart. I should have probably, but at any rate, so then the next year, Eli Lilly, which is a multi-billion-dollar company, they also said they read the paper and they wanted to license it for human health things. And they said the same thing. I mentioned the patent. They said, well, we want to license it. We'll give you a consulting fee, give you another $200,000 grant, and they're going to work on it. And I thought, this is wonderful. But you know what happened? I did get a better car, and I did get the grant to help support some of the students. But those 2 large companies didn't do very much. They tried a few experiments, and they didn't work. And then, like I remember, the year after that, there was a company Sal Snyder started. He's a very famous neuroscientist in Baltimore, and they raised all this money, but they didn't know what to do with it. So I remember they came up, we sent them up, and they said, well, can we license something? And I said, well, what would you like to license? And they said, anything. And I said, well, okay, we developed these new polymers and there's new materials, and we're collaborating on this brain cancer project with actually somebody at Johns Hopkins where Sal was, Henry Brem. So I said, why don't you fund me and fund him? And they said, yeah. And then they hired one of my graduate students, and they worked on this project. And this company had 50 people, but they devoted all their attention pretty much to this one project we developed in the lab. And then they hired my postdoc, and he worked really hard. And I thought, boy, this tiny company, they're doing much better than these multi-billion-dollar companies. So then the next year, some venture capitalists approached me about starting a company, and we did, and that did okay too. So I've just kept doing it. I mean, now we've started 40 companies, and they've really, I'd like to think, have changed the world, not only for my students, but I mean, one of those companies is Moderna, where a number of my students have worked. And, I look at these tiny little startups, and of course, people criticize them all the time. I mean, Moderna, like the Boston Globe, had an article, I think it must have been a year ago, saying this isn't how you do science. I mean, they talk to different supposed experts, with my picture on the front page, which isn't so pleasant. But, I think you get used to criticism over the years. And I think that everybody, at the Koch and myself, I mean, I think the idea of these startups, I mean, you look at what's happened in Cambridge and really the world. And I think that they've really changed the world. And they've changed it for our students, and they've changed it for patients. So I know you're going to hear a lot of great stuff tonight from my three colleagues, and but doing much better than we've done. But it's a pleasure to open this up. Thanks so much.
Well, thank you so much for that introduction. I'm now going to introduce Sangeeta Bhatia, and she's going to tell us about her journey through her entrepreneurship work here at the Koch.
Hello. Okay, now I have to follow the Moderna origin story. It's not easy. Let's see. Share screen. Okay, hopefully everyone can see online. I will proceed unless I'm alerted otherwise. Hi, everyone. It's good to see you. So I'm Sangeeta Bhatia. I'm on the faculty here at the Koch. I'm a physician and an engineer by training. And I wanted to tell you two things today. I have two slides. One is about the power of nanotechnology and cancer. I run the Marble Center for Cancer and Nanomedicine here at the Koch, which is a collection of 6 faculty and 200 researchers. And Tarek Fidel, who's our director, is here in the back if you have questions about it. And as a group, Bob and I and Darrell Irvine, who you'll hear from later, Paula Hammond and Angie Belcher and Dan Anderson, are super interested in the power of the nanoscale. So the nanoscale, just to kind of orient you, a human hair is 100 microns. We make materials that are about 1,000 times smaller than that, so 100 nanometers and smaller. And we have just a whole portfolio of materials that we have developed or have been developed in the literature that can really do interesting and important things for cancer. And so you heard from Bob about packaging mRNA. That's actually done in a lipid nanoparticle akin to a liposome. You can make materials that can image cancer. You can make materials that stimulate the immune system in cancer, and Darrell will tell you about that. You can make materials that can carry therapeutics into cancer, genome edit, and I'm going to tell you about sensors from my own lab. So this is a really fascinating space. If you haven't heard about it, we're happy to share more. We have a website. And this sort of interface of cancer and nanotechnology has led to a lot of companies, not the 40 that Bob mentioned, but many of them here from the KI. So with that as a backdrop, I want to tell you just sort of one specific vignette about the origin story of a company called Glimpse Bio. And we're really lucky tonight. We have the chairman of the board here, Steve Kafka, and our chief scientific officer, Wendy Winkler, and our biology head in NASH, Sophie Casanavi. So welcome, everybody. So this is the sort of just quick origin story. I know we're going to do a panel later of Glimpse. So the story starts back in 2009. And I had 2 graduate students in the lab. One was named Todd Harris, and he would go on to become a serial entrepreneur. He's the CEO of a company now called Tyra Biosciences that makes small molecules for resistant cancer. And the other was a student named Jeff von Maltzahn, who's now a partner at Flagship Pioneering, and he's started a bunch of companies. So these are, actually, we have a lot of special students here at MIT, I will say, but these were 2 real special ones. And in the lab, what they were interested in was developing a way to detect invasive cancer. So you have a cancer and it's invading and spreading, and how do you detect that at its earliest possible point so that you can intervene? And what was known was that cancers, when they do that, they make enzymes that help chew through the tissue. And these enzymes are called proteases, and they cut peptides. And so the enzymes are depicted here as that little Pac-Man, and they're catalysts, so they don't get consumed when they do the cutting. So they can cut many, many, many copies of the peptide. And so what Todd and Jeff had invented was a nanosensor. They made a nanomaterial. This one is about 50 nanometers, so it's even smaller than the ones on the last slide. They decorated it with these peptides, and they showed that if you had one of these proteases, these particular enzymes, that you could cut a lot of these peptides. And they made a version of this material so that when that cutting reaction happened, it would light up. Okay, so it was an ultra-sensitive sensor. So that was 2009, and this fellow here, Gabe Kwong, who was a postdoc in my lab, came from Caltech, where he worked with Jim Heath. Some of you may know, Jim is now the president of the Institute for Systems Biology in Seattle. So Gabe came to work for us, and what I said to him, which is something that all PIs say to incoming postdocs, is I just need you to finish the last figure in this paper. So 4 years later, he published the paper that you see there in 2013. And the last figure in the paper was supposed to be show that these sensors can detect disease in liver disease, liver fibrosis, and in colon cancer. Those were the kind of two applications that we had in mind. And what Gabe realized was that we had made a really sensitive sensor. That's great, but it's actually not enough for detecting disease to have something that's really sensitive. You guys will now all know this because you've been in the COVID world, right? And if you have something that's just super sensitive, you can have what's called false positives, right? And so the way around false positives is to measure multiple things. We call that multiplexing. And so what Gabe figured out was that he could make many, many different sensors. There's 550 proteases in the genome, 2% of the genome. And so we could make many, many sensors, about 20 of them for each disease, and you could profile protease activity. And then you had to make a disease call. Is there a disease or not? And it turns out that's hard to do. And so we use machine learning, which is artificial intelligence, to train the computer to make the disease call when you make 20 measurements at once. Okay, so Gabe invented these multiplex nanosensors in the machine learning. And so then we went on to start the company. So the problem we were trying to solve was to replace a biopsy with a non-invasive tool. We made multiplex nanosensors for protease activity. We published that paper in 2013. And then we needed some funding. And I think you're going to hear later from Angela and Darrell these like gorgeous stories of how they found, you know, a great venture capitalist who could help pull together the early team. But in our case, we actually had to really scrap. So we started with MIT funding from a little accelerator grant, from Frontiers grants. I know that some of you are donors to that program, from angel investors. Our first VC investors were Polaris and Arch. And actually the introduction to Polaris, just to show you how special the KI is, came from Bob Langer. It's a very small community. We were at a football game, that's another story. We got NIH grants, and then we got a pharma deal with Gilead. So anywhere we could get funding for this project, we would push it forward. And the timeline was, so the paper was 2013. The seed investing was, this is a delay. Seed round was closed in 2015. And we still didn't have the executives around the project to really give it a push. Bob was on the board by then of the company, and he turned to me, actually, I don't know if you remember this, Bob, the end of 2017, and said, Sangeeta, why don't you take sabbatical and give it a push? So you remember that? Thank you. So in 2018, I took sabbatical, raised a Series A. We hired then experienced executives, built out the whole team, including Wendy, who you see here tonight, got our Series B lead, which Steve is here tonight. And as of this morning, the company has announced their first patient data in their lead indication, which will be shown in 2 weeks at a liver meeting. And the disease that they're going after first is a disease that leads to cancer. It's called NASH, non-alcoholic steatohepatitis. So that's just a quick origin story. In the lab, we've worked on a lot of different diseases. So we consider this, what we call at MIT, a platform technology. So you can do a lot of different things. And sometimes the entrepreneurship comes in actually making the right choices about what's next. And the students in the lab are all engineers, and they've worked on a whole bunch of different formats of this. So I showed you the light up version. I showed you the light up version of this test, but there's also a breathalyzer version of this test. There's a version of this test that we published this year where you can actually see the tumors with a PET scan. And there's a version that actually carries RNA, so it can detect the cancer and treat the cancer. So there's all different formats. And hopefully we'll be broadening our impact beyond this first indication to many more in the platform. Before I sit down, I want to tell you one the organizers asked us to tell a fun story. So the fun story here is that in 2009, when this invention happened, I took my daughter, who was then, I have two daughters, the 6-year-old, to a meeting where Bob was. And for those of you who know Bob, he has a sweet tooth. And so does my 6-year-old daughter at the time. We met up at the dessert table, and Bob handed her a brownie. And that was the year that we described the very first invention of this technology. So this year, clinical data is coming out in 2 weeks. That little girl just started college. So it gives you a sense of like the dedication of this group of people and what a family we are and how long it takes to shepherd these projects to the clinic. So thank you.
And I'd like to welcome our next speaker, Darrell Irvine.
Yep. Fortunately, I didn't have to follow Bob directly, but Sangeeta's not easy to follow either. So I'm Darrell Irvine. I'm faculty in biological engineering. I've been at MIT almost 20 years. I've been part of 4 different startups, but I'm just going to tell you about one particular experience. And I think a lot of the ideas resonate with things that Sangeeta was saying. And the story, the technology that I'm going to tell you about that got spun out to a company called Elisio Therapeutics was focused on the idea of making better vaccines, in particular cancer vaccines, vaccines that could activate T cells. And just to make sure everybody's on the same page about what's needed, what is a cancer vaccine and what might that entail. This involves injecting into the tissue an antigen that is going to be taken in lymphatic vessels to a draining lymph node. This is more or less what happens with all of the COVID vaccines that probably everybody's had here. And for a cancer vaccine, that antigen would be typically a fragment, a sequence of amino acids, a short peptide that is different in cancer cells from healthy cells, a mutated sequence. And if specialized immune cells in the lymph node called dendritic cells can pick up that peptide, they can present it to a T cell, and if that T cell recognizes that cancer antigen, it can be activated to proliferate, generate a large number of those T cells, and those cells could then disseminate and go after cancer wherever it's located in the body and attack it. And this kind of therapeutic you could envision combining with many other kinds of cancer immunotherapies, such as checkpoint blockade, that might help make those T cells be particularly functional. And the place we started was really a very simple problem, which was how do you get the vaccine to go to the lymph node efficiently? And we started with, I had a chemist postdoc who joined the lab and who started on this project, and every time he would present in lab meeting, he would say, "I'm just a simple chemist." But he had a very smart way of looking at the problem of making a better vaccine, which is he thought about it as a physical chemistry problem. He asked, where do these molecules go and what cells get them? And he recognized that when we inject these vaccines, they have essentially two ways to get out of the tissue. They can either get convected into lymph and enter lymph vessels, which is where we want them to go to the lymph node, or they could get into blood vessels. And if they went into the blood, they just get rapidly diluted and degraded, and you've essentially wasted your vaccine in that situation. And he was shocked by some early experiments that he did in mice, where he found that if he labeled these vaccines to track where they go, that an almost undetectable amount of vaccine was actually arriving at lymph nodes when he made these peptide vaccines that were very attractive because they're very easy to manufacture, very safe, and very scalable. And so as we dug into this problem, we realized that there's a really fundamental piece of biology that was governing what was happening. And that's when you inject something into tissues, to first order, the outcome of where that injected material is going to go is governed by molecular size. So if you're small enough to get through the tight, the basement membrane and tight junctions of the endothelial cells that wrap the blood vessels, then you're going to go into the blood, because the blood clears tenfold more fluid per unit time from the tissue than the lymph does. But if you're too large to diffuse through these basically the seal that lines the blood vasculature, then instead you'll go with the convecting lymph and get carried into lymph vessels and then go to the lymph node. And in fact, you can measure, you can inject material into the skin or into the muscle and then measure how much of the injected material goes into the lymphatic vessels, and you get a graph like this where with increasing molecular size, and this is just a proxy for, increasing molecular weight, which is just a proxy for molecular size, the bigger you are, the more of the material you inject shows up in the draining lymph. And there were two things on this graph that immediately caught our attention, which is first, that our peptide vaccines that were a few kilodaltons in size down here at this end, well, this explained what was happening to them. They were all being injected and going straight into the blood and being lost. But this graph also suggested a solution, which was if you're large enough, about 4 or 5 nanometers in hydrodynamic size, then you're large enough that you're trafficking one way from blood to lymph. And we further realized that the most prevalent protein in your blood, albumin, is one of these molecules, that it comes out of blood vessels and it goes through the tissue and exits through the lymph. It's sort of a continuous conveyor belt of material trafficking through there. And so that inspired the postdoc Hai Peng to create these molecules that would use albumin as a built-in chaperone that already exists in the tissue as a way to send vaccines to lymph nodes. So he came up with this very simple but powerful idea that you'd take an albumin binding lipid tail, because one of albumin's main jobs in vivo is to ferry lipids around as a nutrient. And so he made these molecules where he'd link his cancer antigen to a short polymer spacer, and then this lipid that went on injection would bind to albumin that's already in the tissue, and then albumin would carry it to the lymph node. And what we showed, and he carried out a whole exhaustive set of biochemistry studies to show that when you do this, you could take a molecule that was essentially undetectable in the lymph node and show that it robustly accumulated. And then if you ask what this did to the vaccine response, you could show that you could take cancer antigens that when you vaccinated and then tried to measure how much of a T cell response was generated, you could have antigens that initially you couldn't detect hardly anything, that could be increased, 20 to 50 fold, but it's more like from 0 to something for a variety of different cancer antigens. And that then would allow you to take a vaccine, and this is a melanoma model in mice where he showed that a traditional peptide vaccine in red couldn't change tumor growth at all compared to untreated, but when he optimally delivered the vaccine to lymph nodes, now you had a vaccine that was doing something. And so that set of data that was published, and the key findings were in this Nature paper in 2014. And this was the first time in the lab where I said, this is something that we should really try and spin out. And this was my first experience doing a startup. And so there were things that we did well and things that we did poorly. This was a case where a serial entrepreneur who had worked with faculty at MIT before came to visit me. He got interested in the story. He signed on to help start the company. And so we launched Elisio. And there were multiple things that sort of slowed down the progress. We didn't raise enough money initially because I didn't know how much money we really needed to raise. And the initial leadership was not the right set of folks to make good decisions early on. But some really good things happened. And one of those was that the best PhD student that I ever had, who did not work on this project, but he knew all the science, he went to the company. And that was a really transformative decision because he knew how I thought about the science, and he was able to present that to anyone and everyone. And as with many startups, this junior guy who just earned his PhD was presenting to heads of research at big pharma, presenting it to many, many VCs to raise money, and really working beyond his years. And so the company eventually righted itself and was able to hire a really outstanding CEO and realigned their board in 2018. They chose their strategy. They decided to go after a setting of minimal residual disease where you would have patients with cancer where their tumor had been resected, and you would try and prevent relapse to try and put yourself in a situation where the immune system has a better chance. And so that led them in 2021, Elisio raised a $73 million Series B that allowed them to push their initial program into the clinic. And they've just treated their first patient at the beginning of October. And so, this has been a long journey. The paper was published in 2014, just seeing a first patient dosed this past month. But I think it really illustrates the long road it sometimes takes to get things out of the lab. And in this case, that we're mostly governed by things other than the science. The good thing was the science really worked. And when it moved to the company, they were able to show in many settings that this idea was general and could be used to make better vaccines. But now I think the exciting opportunity is presented by COVID is the potential of vaccine technologies like this to be used in many settings and beyond cancer, including infectious disease. I'll stop there.
Thank you so much, Darrell. And it's my pleasure to introduce our last speaker, and then we'll move over to the panel discussion. And our last speaker is Angela Koehler.
All right. So it's a pleasure to be here. I want to say hello to the at-home people through that camera right there. I really am just a simple organic chemist, and I'll tell you a little bit about some of the stories in our lab that led to a company called Kronos Bio that's represented here. So in our lab, we're very much interested not so much in drug discovery, but chemical probe discoveries. We're interested in our colleagues across the street at the Broad Institute, colleagues all over the globe that are using human-guided biology to nominate potential therapeutic targets. And so our lab is interested in trying to come up with ways to pharmacologically validate those targets or potential novel mechanisms to think about how to develop drugs or potentially lower the activation barrier for our colleagues in industry to go after certain types of drug targets. And so we really have an interest in looking at these lists, building these chemical probes, and then trying to understand what the impact of modulating those targets are in a physiologically relevant setting. Every once in a while, well, I should say the vast majority of the compounds from our lab really stay in this tool compound bucket. But every once in a while, lightning will strike, and we'll look at these molecules and think that they may have the potential to actually serve as a lead molecule. And that's a combination of logic associated with biology and the function of those molecules, as well as their chemical architecture. So 2 molecules that fell into this bucket of potential lead structures came to fruition in my third year here as a faculty member at the Koch Institute. KIMS 2008 and KARV03, we call CARV3. KIMS 2008 is an interesting molecule because it attenuates MYC-driven transcription. MYC is deregulated in about 70% of human tumors, and the thinking is that if you could come up with an agent against MYC, you could take out a broad swath of tumors for which MYC is a driver of the oncogenic, the malignant phenotype. This molecule acted through a novel mechanism. It binds to a protein that interacts with MYC called MAX, and what it does is it reduces MYC-driven transcription, and it actually degrades MYC itself. We had a lot of interesting data around this molecule in 2017. We worked with colleagues at the Stanford School of Medicine to generate preliminary in vivo data in a couple of different settings. But what we did not have in 2017 was sort of a detailed medicinal chemistry map, PK/PD guided medicinal chemistry, and we had incomplete knowledge of the pharmacology around this molecule. CARV3 is a molecule that inhibits androgen receptor-driven transcription. It works indirectly. It's actually an ultra-selective CDK9 inhibitor. There was some surprises in this story. It modulates ARV7 levels in cellular models of enzalutamide-resistant prostate cancer. This molecule arose through a partnership with Steve Balk's group across the river as part of the Bridge project that is meant to nucleate partnerships between MIT and Dana-Farber. Moreover, Marius Popp, who's here in the audience, discovered this molecule in partnership as part of the Janssen Transcend project, which was in its final year when I joined the Koch Institute. But again, we had limited medicinal chemistry information, limited pharmacology around this early stage compound, and at this point we had no in vivo proof of concept data. So in academia, the thinking is that sort of the logic for running a lab is that when you think about medicinal chemistry, it's one postdoc per molecule. In our case, it wasn't quite so bad. For MS2, we had Francisco Caballero, who was involved in the original discovery of this molecule. Eric Stefan was our medicinal chemist and trying to establish some structure-activity relationships. And later on, Nick Struntz joined to understand some of the biochemistry. But it was clear that we were going to be somewhat limited in the numbers of molecules that we could make. And then for CARV3, a somewhat similar situation. Marius actually discovered the molecule. Dave Freeman, who is also here, was our medicinal chemist and trying to make some optimization for the molecule. And then later, Andre Richters joined the team and also thought about some of that biochemistry. So at this stage, we were running out of money for the CARV3 story because the Transcend project had ended and we had not written any grants to think about how to advance this molecule into the necessary studies. For the MYC molecule, we were generously funded by MIT's Deshpande Center to start to check off some of these boxes with contract research organizations to try and understand the pharmacology a bit better. So one thing that happened that was transformative in late 2016, my colleague Rick Young across the street at the Whitehead Institute hosted a meeting on around drugging transcription. I gave a lecture talking a little bit about this molecule, MS2008, didn't show the structure, but talked a little bit about the results we were seeing in vivo. And then all of a sudden, I had requests from many venture capitalists to think about this particular molecule. So one individual who came into my office is Chris Wolfong, who's sitting right here, representing Two River, which is a company that's focused around building biotechnology life sciences companies. And this is the team at Two River. They've been involved with a number of well-known companies. Chris and I think after many whiteboard sessions sort of clicked in terms of our vision around not just advancing the molecules that I've shown you on these slides, but also thinking about the platform that gave rise to these molecules. So that gave rise to a company called Kronos Bio. We took the novel compositions here from MIT and we brought in IP from my previous life at Harvard, including the platform technology called the small molecule microarray. And I was a scientific founder. Chris came in as a founding COO. Marius was the director of biology. Dave was a chemist, and now he has a fancier title. And I should say, now that the company is sort of well on its way, Chris and Marius have moved on to a new company that is connected to research here at the Koch as well. So maybe someday we'll hear more about that. And I should say, Marius and Dave often fight about who is employee number one. I think you're 1A and you're 1B. I should say, it is, as all the other speakers have said, it's absolutely critical to have people who know the technologies and the molecules come into the company at an early stage. So this is the timeline for Kronos. We started in 2017, brought in executive leadership from Two River. We had a seed round of $18 million in 2018. We continue to advance these technologies and molecules that I showed you about. We had a Series A of $105 million led by Vita Ventures and Omega Fund. And then one cool thing is that we had the opportunity to bring in assets from Gilead, a Syk inhibitor portfolio that was very consistent and highly aligned with some of our interests in the AML space. None of that would be possible without, of course, one of the most transformative events was recruiting Norbert Bischofberger, who had previously been a CSO at Gilead, to come be the CEO of Kronos. We also had the opportunity to bring in amazing talent such as George DiMartino, who's the Chief Medical Officer, and Chris Dinsmore, who's the Chief Scientific Officer here in Cambridge. I should say this company is split between San Mateo and Cambridge. And then finally, just over a year ago, the company transitioned into the public space with an IPO. Moving forward, the company is now advancing the Syk inhibitor portfolio in AML, moving towards phase 3. The MYC molecule that I showed you, it was deprioritized for a variety of reasons. It was an example where in a commercial setting, when you had all of this professional talent at the table, it became clear that there were certain liabilities with that molecule. And so we went back to actually think about new screens and new strategies to develop new agents around that specific target. But that CDK9 inhibitor, CAR3 that I mentioned, Dave Freeman and colleagues made hundreds of analogs and advanced that to an agent that is being studied now in clinical trials. And then the team, of course, is using the platform that we developed to look more broadly against a set of oncogenic transcriptional targets. And so with that, I've also started other companies in the past. This has been a real pleasure. For me, it's been an example of at least a success story so far. I have no regrets, and I continue to interact with my colleagues, and hopefully this is an example for others at MIT to think about a similar path. So with that, I'll end. Good.
Yeah, this is where we do the stage management now. I'd like to welcome all of the panelists up to the front. And firstly, I want to thank you all for giving your viewpoints of entrepreneurship here at the Koch. I would like to encourage our Zoom audience to participate via the Q&A session. But I'm going to kick off with the first question while folks are getting their mics on. And that is, how do you decide which idea to move forward? And Angela looks like she's ready to answer that question.
Well, in the case I just gave you, it was necessity. We really didn't have a clear path at MIT, the resources, the funding to move forward within this setting. If we decided we were going to make 300 different molecules here and evaluate the pharmacology, we would very quickly overwhelm MIT's infrastructure, I think. So for us, it was certainly a necessity.
Well, for my case, I think one of the things of going through the experience of starting something up, you learn more about what it takes to see something get out of the lab and survive. So I think at this stage, we sort of know when we've tested it enough in the lab. And I've certainly learned that the more we test it in the lab, the better. If you move things out too early, you can discover things very quickly that you didn't know that are going to kill it going out of the lab.
Yeah, I would agree. I think the other thing is for most of these projects, there's a regulatory conversation to be had. And that's not really graduate research, right? To talk to the regulators, like often you need 2-species toxicology or you need scale-up manufacturing. And it's just, the kind of thing that there are professionals that can advance more quickly. And so that's one of the reasons I think to move on and move it forward to patients.
So I mean, of course, I've been doing this a long time, so I've had a lot of failures and some successes, and so kind of my thinking's evolved, and I hate failing. It's just, I mean, because whether I think about it academically or from the companies, I feel like if we're successful, then people will keep giving you more funding to do more good things. So the formula, like I've kind of developed after now close to 50 years, is that you. And medicine takes a long time. I mean, I've done some non-medical things too, but medicine takes a long time. So the kind of formula that I figured out, I mean, not that it's right, but is that you have, like a big idea that's what I call platform technology, something that can be used for many different applications. It's sort of a plug and play kind of thing. That you have a paper in a really top journal, and some of this has been mentioned by others, but journals like Science or Nature, that you have really good patents, and and for me also that I have students who have spent a lot of time on this that really want to see it make a difference in the world, that want to go work at those companies. So and finally, I guess that you have like an in vivo proof of principle, again, because it takes such a long time.
Great, thank you. I do just want to warn everybody, I think we're going to run over by a little bit. So feel free to stay or feel free to leave if you have a time constraint, but I think we've got a lot to talk about this evening. So a question that's just coming in from Ellen Clegg, who I actually know from my days at the Broad. There's a saying amongst digital entrepreneurs that fail fast. Does that apply to the life science field, especially where regulating agencies and human subjects might be involved?
Okay, well, I'll try to answer first, and most of the world will probably disagree with me, but I don't agree with that. I mean, almost everybody says that's what you want to do, but I think it's wrong. I'll pick nanotechnology, which is at the heart of some of this, as an example. I mean, if you have a platform technology, then it's like, and again, people have to realize, and I'm sure you do, that medicine is limited. Like you get a couple shots on goal. If your first clinical trial doesn't work, you raise so much money, and either you could succeed wildly or you will not succeed wildly. So the problem is if you pick your first clinical trial wrong, which could be a business reason, it could be an FDA reason, then you will fail. And somebody could say the technology wasn't good. So I'll give you my personal examples. So, we started a company called Bind. And I remember and basically we developed nanoparticles, what I talked about. And actually we did pretty well in some ways. We raised some money, we got into clinical trials, but around the same time, immunotherapy came along, and that was better for that did better than some of the things that we were working on. So, and also, and this relates to one of the things that Darrell said, we didn't hire great CEOs. And so one of them, we had CFOs, one of them took a bad loan. And when the clinical trial it actually worked okay, but it didn't work as well as immunotherapy. So when that happened, the company Hercules that gave us the loan, they called it, and the company temporarily went bankrupt. Ultimately, we sold it to Pfizer. And there were all these articles in Nature and other places that said nanotechnology doesn't work. Well, now you come a couple of years later and you look at what we've done at Moderna and what other companies have done with nanotechnology, and all of a sudden nanotechnology, I mean, every time the particles are different, of course, but it doesn't really matter. It's the principle that's key. And if you pick the right application, and the right clinical trials, it can make all the difference in the world. So like I say, almost everybody that you talk to says failing fast is good. I just think if you have a platform technology, it's absolutely the wrong way to look at it. And like I say, I'm probably almost one of the very few people that would say that.
Any other comments?
It's hard to add anything to that great answer.
But all the things that everybody's doing, I mean, you know, you look at what everybody's talking about, in some ways they fit the same thing, you know, and if they're and medicine is just so expensive, you know, you takes billions of dollars to do one thing. And, you know, you looked at what, you know, what Angela just said, and, you know, it's, you know, it's just huge risk, but also huge reward.
I do want to just remind the in-person audience that you are more than welcome to ask a question, and Erica has a mic, so feel free to join in person.
No, I was just going to add that I think one thing that's I think we all have in common as engineers and chemists is actually having an invention and finding the right application. So we all care about cancer, but actually cancer is a very broad swath of problems. And so it's often the case that, you know, in our case, we had a sensor. Is that an early detection sensor? Is it a recurrent sensor? Is it a drug response sensor? Is it a sensor outside of cancer, which is actually where it's going to go first? You know, that's true for every one of these projects. And so there's, you know, I think Bob's point is very well taken. It's not so much about failing in a particular indication. It's actually about finding the right use case for your invention.
So one thing I was going to say is that in our field, we like to kill molecules quickly. But what I worry about is that people will then kill a target quickly. And I think that would be a big mistake.
I had another question that's come in from our Zoom audience. How important are patents and different intellectual property rights, and how do they contribute towards forming a company within life sciences?
Well, Bob teaches the intellectual property lecture in our course. So
I'll okay. Well, I think Angela's volunteering me, but but I mean it's. I mean, in life sciences, it's absolutely essential. I mean, we have investors here and other people. You know, the problem is again, it goes to what we said before. I think Tufts estimates it costs $2 to $3 billion to create a new drug, and you know, investors can invest in anything they want, and you know, which means they could invest in a Facebook or they could invest in a Moderna. But if you didn't have intellectual property, nobody would invest in a medical company because it costs billions of dollars, and then anybody else can do it. So to me, if you, when I hear that people think you should give intellectual property away, I mean, the one thing you can be sure about is there'll be a lot more Facebooks and a lot less Modernas, and you can imagine the effect that'll have on the world. So I just think intellectual property. I mean, I can't imagine any biotech investor invest. I mean, like I say, there's a lot better investors, you know, that know a lot more than I do, but I just don't know anybody that would invest without doing really due diligence on intellectual property. And I know everybody up here, you know, and I think one of the things that Koch's done well is file for good intellectual property and start good companies from it.
I'm actually going to make a leap from your last comment about investors. Can you all please share your thoughts on how you chose your early stage investors, your first investors? How did you go about doing that?
You guys go first.
It's not that we always have the
choice,
the luxury of the choice, but I think when you do have the choice, there are very different mindsets. There are people who want to build a company with you. There are people who want the fastest return on investment, and you have to be aligned on what the goals are.
I totally agree. We could have worked with many different groups in the earliest stages of thinking about the right way to advance these molecules and platforms. But, you know, again, I think a lot of it was mojo with Chris, being able to go to the whiteboard, feeling like we could disagree, but also come to agreement. And we were highly aligned in terms of the vision.
Yeah, I would agree. I think in this case, we were, at least with Glimpse, we were going feel like maybe every one of my companies, we've been trying to do something brand new, which is exciting but hard. And so collecting investors that have different experiences can be really important. So for example, with Glimpse, we had Polaris on the East Coast and Arch, which was really very different. At the time had just funded Grail, which is a liquid biopsy company. And so we kind of really deliberately tried to gather those perspectives as early investors to give us kind of the biggest perspective about how to take the technology forward, and also the best networks for talent and other things.
So taking that leap from selecting an investor, you all touched on how important it was to form a really strong leadership team. How did you go about doing that? Where did you find these people? Without embarrassing the folks in the audience.
We had a member of our investment team, I guess it was luck and just a conversation with Norbert, and then sort of a dating period. We got lucky. It was a huge moment to bring in someone who had spent a career at a company like Gilead, and to completely transform the way the trajectory of the company. Yeah. But I think there was some luck there.
I will say there's so we used to have a TLO director named Lita Nelson, and some of you in the crowd will know her. And when I first came to MIT, it was 2005, and I was starting my first MIT-based company. I went to her and I said, I'm thinking of starting a company. How do I, find my first CEO? And she said, really unsatisfyingly, oh, it's the ecosystem. I said, what does that mean? She said, you put it out in the air that you're thinking of starting a company, and you go to these functions and you talk to people. And at the time, an engineer, I thought, like, there must be a better system. She must be wrong. It turned out she's totally right. Like, that's actually how it happens, is you talk to somebody, or you're at an event, or it's, and then lightning strikes. And there are also, recruiters and other things that you can use to formalize. But for the most part, it is the ecosystem, I think. Yeah. Maybe that's like special KI sauce.
Yeah. Well, and also, it's your network, and this is something that I'm particularly passionate about, and how important it is for us all to build a network of great people and great leaders around us. I'm going to jump into another question that's come in remotely. What are your thoughts on starting a biotech that's directed at diseases that impact less wealthy regions of the world, for example, malaria, where investors may view the potential return on investment as not being as profitable as something like cancer? And I'm actually going to point this to Sangeeta, because I know you and I have both talked about this. So.
Yeah. That's an important question. Yeah. So you can all tell I'm of Indian descent, and I, grew up summers in India, going to my aunt's clinic, seeing, what healthcare is like in other parts of the world. And so I've been very interested in global oncology for a long time. I gave a TED talk about this Glimpse technology, and it's actually about like the vision that you could have a paper diagnostic in a low resource setting to diagnose cancer. And I still actually really believe in that, although it's not a good business plan for the beginning. And so I think that in the academic setting, you actually can continue to push on that long-term vision. And you have to kind of work multiple threads to get the technology out there. You also have to be realistic and recognize like what is a profitable first investment, and consider innovative business models. And actually, Bob is a great example. I mean, you're working on micronutrients, amongst other things, in a totally different business model. So I think, whatever shots on goal you have, to keep that in your mind's eye.
We actually have a question from the audience, and it's from Matt van der Heijden.
So first of all, thanks for doing this, guys. And I wonder if you could comment a bit on the role of the trainees. That many of you brought this up during your talk, but how do you guys think about getting trainees involved in entrepreneurship? Many of them are interested in it. Some may get lucky. Any thoughts on that, I think, might be appreciated.
It's never been a huge issue in our group. I think a lot of the folks who join our group have an interest in trying to translate their ideas to the wider world via either new venture creation or other mechanisms. But I think before the pandemic, we tended to have a cohort that was certainly interested in moving towards this academic sector. I think many of our trainees, in part because of the innovation associated with the pandemic, have decided that actually industry is a better place to be innovative, and they're less interested in academia. I actually view that a bit as that's a problem for us, by the way. But in our group, it's never been a challenge.
Yeah, as I said when I was speaking, having great people from the lab go to the company has made a huge difference the couple of times it's happened for me. And I can remember years ago when I was a junior faculty, Bob giving a talk to the faculty about what works and what doesn't work with translation. And he mentioned that a lot of companies fail because they didn't get good people. It has nothing to do with the science or the ideas. And I really took that to heart. I think it's absolutely true with startups because you're really hinging your success on a handful of people.
I'm going to make a comment actually, Matt. As you know, as the new person to the Koch, one thing that has really impressed me here is the entrepreneurial spirit is very much throughout the entire institute. You know, I've seen that, you know, in other places and other institutes, it tends to be reserved at the C level, and I just don't see that here at the Koch. I see this entrepreneurial spirit actually all the way through the institute. I think, you know, the postgrads, the postdocs, and the trainees, everybody is really thinking about this, and that's something that I think that is particularly wonderful about being here. Talking about the being an academic, how do you balance your time between the startup, your academic responsibilities? Some of you have clinical responsibilities as well. How do you juggle all of this? How do you focus on your priorities? You just do.
I think we all have different strategies. I think Angela and I were looking at each other because I have to say she's the mother of 4, so it's just incredible, like what she's juggling. But I think, you know, I personally, I compartmentalize and I, you know, collect great people, which is, I think, to help. I know everyone has their own strategies. Bob, for example, is the quickest responder ever to any email ever, and so he never. I think that's like. I think it's everybody. So I think we all have our different ways.
Yeah, I can back that up. He is the fastest responder to email. I mean, do you have your startup brain and your academic brain? Do you switch from one side to the other? Is it all just one big mess and you have a big to-do list?
So my husband was a postdoc in Bob's lab, and my husband's a real pain in the ass. Like, you should start this company, you should start that company, because he trained with Bob. So I have no choice.
I think the thing is, you know, you could put anything in that box: working on the company, writing the rec letter for your postdoc, you know, teaching class, leading the lab. I mean, it's writing the grants. I don't know how any of us manage it, but.
I will say that, you know, I think there's amazing amount of multitasking, although somebody on that table over there is going to tell me that multitasking doesn't actually exist. Actually makes, you know, a lot of the magic and a lot of the energy that happens here at the Koch and also at your startup. I think this flexibility, this, you know, this flipping back and forth is actually what creates a lot of the energy here. So I actually think it's a great thing. Any other questions from the audience? Yes, we have one at the back.
Hi, hello, good evening. It's Emiliano from Novo Nordisk. My question is, has the role of big pharma changed over the years in relation with startup and the way they may assist them? I mean, you, Bob, were talking in your examples of an interaction that was felt pretty much transactional with them. They wanted a piece of intellectual property, they interacted with you. Has this changed, or you, younger entrepreneur, you see that pharma can play some roles that are not just transactional, but also helping to pour some experience into some enthusiastic people that have a company in mind?
I think that some of the most interesting projects in our academic group are equal partnerships with colleagues in the pharmaceutical industry that have a common interest in going after challenging recalcitrant targets. And we're excited to solve bottlenecks. And this goes back to something that Matt asked about. They're interested in working with us in part to help raise the next generation of pharmaceutical researchers. So we've really enjoyed at least the academic industry relationship. And I think we're taking on fairly challenging problems.
I have another question that's just come in, which I think is a super interesting one. So you all mentioned how you had angel investment in some of your projects. Given that it takes $1 billion to develop a drug, is there a place for an angel investor in this field, given that they have much less money and a much shorter time horizon? Sangeeta?
Sure. Yeah, I think it depends on the project and the angel investors. So it's in Glimpse, the 2 angel investors were people that I knew incredibly well that had specific domain expertise. So one of them was a West Coast tech VC. So she knew West Coast venture capital, and she was somebody I trusted incredibly and who trusted me. She was my roommate from college. Her name is Teresa Gao. She's a Midas investor. And I'm super proud of her. And then the other one was somebody I met through the Coke, actually, Kiran Mazumdar-Shah, who was the founder of Biocon. And I developed a relationship with her because she actually funded a postdoc of mine to go back to India to become a professor. And so it's worth noting these are 2 women believing in a woman, and almost with no business plan, just said like, go for it, we'll seed you. And so, I mean, there's I think definitely a place for that kind of angel investor.
Another question that's come in virtually. Does anybody in person? Nope, I'll take a Zoom question. Would be interesting to learn your advice for a fellow MIT alumni who are not currently part of the MIT ecosystem today. How can they get involved and use some of the case models that we've talked about, that are not necessarily new platform technologies? How can they help leverage some of these technologies? It's a pretty broad question.
I'm not sure I fully got what the question is.
Yeah, I'm not sure. I agree with Darrell. I think it.
How can they get access to these platforms?
This is. I'm still not sure I understand the question.
So they're outside of the Coke, they're outside of the MIT platform. How can they get involved in and leverage these technologies? How do they go about working with and licensing some of these technologies? Which is a whole IP and licensing class in itself.
I mean, I think there's a number of mechanisms. So one of them is just forming connections at events like this, the With Insight series. Obviously, the Technology Licensing Office has all of our IP, available to them. And then we have various kind of, if they're an industrial affiliate programs. So the Nanomedicine Center has one. There's a variety around campus. So I think there's sort of trying to figure out how you could dock into MIT.
Yeah. And I think it's really a question around how to navigate building a relationship around the technology platforms that we have here, working with the industry liaison program that we have in MIT, working directly with myself as the executive director here, working with Tarek, who's building a lot of relationships within the nanomedicine groups. So, well, I think we are going to, oh, wait a minute. I've got one question that I really want to ask. Sorry. And then I'm going to wrap up, I promise. What do you think is currently the most awaited breakthrough in medicine and biotech? And it's probably a whole bunch of people.
I still
writing
think it's
checks.
drugging Mick.
Not that she's biased.
Yeah. I didn't talk about it tonight, but I'm very excited about cells as living medicines. And I have a new startup, the CEO's here tonight, where we're trying to do that for the liver.
So, well, if you're looking at me, so I've been asked that question a lot. And, you know, for all the companies we've started, I'm a big believer in basic research. And my answer is always, it's something that doesn't even have a name yet because it hasn't been discovered yet. And an example I often use is CRISPR. You know, if we asked anybody 10 years ago what CRISPR was, they wouldn't know. I mean, and so I think that so much of it just develops by curiosity-driven research, or research that doesn't, you know, research is such an unlinear thing. Like I was telling you about mine. I mean, we started out doing cancer research, and we still do, but that would lead to nanoparticles, and that, of course, you know, would help pave the way for some of the things that we're now seeing with COVID. I mean, who the heck would know? I mean, research to me doesn't work by necessarily thinking that you're gonna, but so many times when you really look at what the breakthroughs are, they're not because somebody intended to. And yet when they develop, you know, and that's the beauty about entrepreneurship and all these things, the industry can capitalize on them and make therapies that can change the world. So I always say, like I say, it doesn't have a name. That's just my 2 cents.
Right, well, that's a great closing statement. I want to thank you all for joining us in person and virtually this evening. We're actually recording this, so if you enjoyed it so much, you can go back and watch it again. We'll look forward to seeing you again at the next session. And I do want to thank all of the organizers who really, really helped make this happen this evening. So a big thank you to you.